my heartfelt desire that we hold this meeting in person and we get to see you all face to face. But as you all know, COVID has made travel very difficult this year. So my hope is that we will conduct an in-person meeting next year and see you all in person. But for today and for tomorrow, we will interact with you virtually, and we hope you will enjoy the meeting proceedings over the next 2 days. And before we go further, I would like to remind you all of a few logistics and housekeeping related issues before we go and I talk about that in detail about the agenda of the meeting. The most important thing is that this session is being recorded. All the meeting proceedings, including the videos, will be put on the CPATH INC website. This website currently already holds the detailed agenda of the meeting as well as the speaker bios and all other on-demand resources. And we encourage you to go to this website and take a look at that. Another point is that in case, which I really do not think will happen, but just in case there's any platform outage, WebEx suddenly shuts down, or some serious technical difficulties happen, we have a backup plan ready and we will email you with instructions. And in case something like that happens, please be on the lookout for an email from us. Another very important point is that or there are going to be at least a few pre-recorded videos that we will be uploading onto the WebEx platform for broadcasting. And the audio for those videos is only available if you are using your computer audio to listen in, not through the phone audio. So as a best practice, I advise everybody to log into this meeting using your computer audio and not phone. Another important point is just to ensure that there is seamless connectivity with WebEx. Please do not use any VPN connection when you're connected to the WebEx or when trying to connect to the WebEx. It just causes issues. So recommend do not use VPN. Another point is that we would obviously welcome if you share our event and INC on social media, Facebook, LinkedIn, Twitter, whatever. So please do share information about our event on your social media posts. And if you have, one of the points, important points of this meeting is that we would welcome feedback from the audience for everything that we are going to be presenting today. So we encourage you to ask questions, and there's a QA panel on the bottom right screen, and please enter your questions in a written format and direct them to all panelists. And now on to the agenda of the meeting and what to expect over the next 2 days. As I said, this is a 3-day session, 3-day event that is spread over a period of 2 days on 19th and 20th of October. The first session, that is the one that is happening right now, we will have lectures and discussions focused on cell and gene therapies in neonates and the landscape of sponsored cell and gene therapy trials in the neonatal population. We will start with a plenary speech by Dr. Jeffrey Siegel from FDA, which will focus on drug development tools to facilitate drug development in the neonatal population. Then there's going to be an award ceremony to Dr. Susan McCune, followed by lectures on cell and gene therapy, and then there's going to be panel discussion. After that, we will take a short 45-minute break, followed by a discussion of and a presentation on INC work group updates. After that, we will take a longer 4-hour break, and then we will reconvene at 6 p.m. Eastern Time to continue. We will reconvene again at 6 p.m. Eastern Time to continue the discussion left over from the morning session. This evening session will focus on diagnostic technologies for whole genome sequencing in newborns, and then ethical considerations related to cell and gene therapies. Then there's again also going to be a panel discussion at the end. And then finally, on day 2, that is tomorrow, October 20th, we're going to be focusing, the session is going to be focusing on real world data and how real world data and its application can transform drug development in the neonatal population. And that is all about what I wanted to talk about the agenda today. But before I go on, I also wanted to again point out that this again detailed agenda and all speaker bios are also available on the CPATH and INC website. And now, without further ado, we will start with the session today. I'm very honored and privileged to welcome Dr. Jeffrey Siegel to deliver the opening plenary talk today. As an introduction, Dr. Siegel is the Director of Office of Drug Evaluation Sciences at FDA, and he will talk about the application of drug development tools and how these drug development tools can facilitate drug development, especially when it concerns to the neonatal population. Now, this topic is very important simply because the ultimate goal of everything that we do at INC is to create drug development tools for the newborn population. So, welcome, Dr. Siegel. Thank you so much for agreeing to give a speech to the INC audience. And I will give you the mic now.
Thank you very much, Kawaljit. And good morning, everyone. I'm delighted to be here to talk to you about drug development tools. Here are my disclaimers. I do have one additional disclaimer, which is that I am not a neonatologist. I'm an internist. I've looked at many presentations from neonatology meetings, and I just find the pictures of the babies so adorable. I wanted to include them, but I'm afraid I don't have the expertise, so my apologies. So here's what I'd like to cover during my presentation. I'm going to talk about the urgent need to develop new tools to facilitate drug development. Talk a little bit about the FDA process for regulating these tools, and talk about a couple of tools in particular, starting with biomarkers, talking about digital health technologies, real-world evidence, and then at the end, talk a little bit about public-private partnerships. So I think we all know that drug development can be very cumbersome and costly. And there have been many failures of drug development programs throughout drug development. And tools can help improve efficiency and reduce the cost. There are unmet medical needs that need to be addressed in rare diseases and pediatrics and other areas, and drug development tools can help with this. We need to incorporate novel data sources beyond the traditional clinical trial data sources. And precision medicine is an important area where biomarkers can help make sure that we get the right drug to the right patient at the right time. And then novel technologies can allow better assessment of clinical responses, and in some cases permit decentralized trials, which are quite important. So in traditional drug development, you proceed from phase 1 to phase 2 to phase 3, and typical study designs can be fine when you're talking about common diseases. But this traditional stage drug development doesn't always work for small populations. There's increased interest in use of real-world evidence to supplement the information that comes from clinical trials. And then advancing technology provides a number of different approaches to development, which includes digital endpoints, decentralized trials, and as I mentioned before, biomarkers to achieve personalized healthcare. So FDA has recognized this need for modernizing drug development and has spent some time diagnosing the problem, figuring out what needs to be done, then designing a new process in FDA, and then spent from 2018 to 2020 implementing this. And one of the important ways that this took place in FDA is in the Office of New Drugs. A new office was set up, the office that I lead, the Office of Drug Evaluation Sciences, which evaluates drug development tools across therapeutic areas. And in particular, my office is responsible for qualifying new tools, in particular, biomarkers and clinical outcome assessments. So I'm going to start by talking about biomarkers. Let's begin with the definition. A biomarker is, as many of you know, a defined characteristic that's measured as an indicator of normal biologic processes, pathogenic processes, or responses to an exposure or intervention, including therapeutic interventions, but not confined to that. And importantly, a biomarker is not an assessment of the way a patient feels, functions, or survives, that's a separate category of clinical outcomes. Congress set up legislation to guide the qualification process for biomarkers. And the process is shown here. It begins with a letter of intent from the group developing the biomarker, then continues on with the plan for qualifying it, and then the full qualification package. And if the data are supportive, the biomarker is qualified for a particular context of use across drug development programs. It's important to recognize that in addition to the biomarker qualification program, there are other ways that biomarkers gain acceptance in drug development. One is, of course, through scientific community consensus based on data. Another is that individual pharmaceutical company sponsors can develop biomarkers and incorporate them into their specific drug development programs as part of the IND process. So these are all separate, but they do interact in important ways. And that's why I've shown it as a Venn diagram. For example, sometimes a biomarker can come in for a more easily qualified use, such as a prognostic biomarker. And then it gets qualified for that. And then it's incorporated into drug development programs, into clinical trials. And then you get the data that show that it's not just a prognostic biomarker, but maybe it also has data to support its use as a surrogate endpoint. So the drug approval process, the IND process, interacts with the biomarker qualification program and scientific community data to advance biomarkers in many important cases. There are a large number of different types of biomarkers, and the different types of biomarkers are summarized in the BEST document. It stands for Biomarkers, Endpoints, and Other Tools. And this can be found on the NIH FDA biomarker website. Here are listed some of the different types of biomarkers. And it's important to recognize the differences between these and the different applications. So there are risk biomarkers. There are biomarkers that are used to monitor the disease over the course of time and to see the effect of therapies. And then all the way up to surrogate biomarkers, pharmacodynamic biomarkers, and other types. This scheme illustrates the different ways that biomarkers can be used in different contexts, depending on where the patients are in the course of the disease process. So before patients may develop the condition, there are risk susceptibility biomarkers. They identify people at greater risk of developing the disease. And later on, when patients develop disease, you can think about a variety of different types of biomarkers, including diagnostic biomarkers, monitoring biomarkers, and prognostic biomarkers that are particularly important because they identify the patients most at risk of progressing. And by enriching clinical trials in the patients most likely to progress, it can increase the likelihood of success of treatment with a therapeutic agent. And I mentioned before surrogate endpoints that are particularly important because they're endpoints that are so tied to clinically meaningful endpoints that they can be used for approval of new drugs. But a lot of data is required before a biomarker can be used as a surrogate endpoint. Biomarkers can help drug development programs in a number of important ways. They can reduce the size and length of pivotal trials. For example, as I mentioned before, diagnostic biomarkers for enrichment. And an example is lupus nephritis, where back before biomarkers were accepted, progression to end-stage renal disease or doubling of serum creatinine was used as an endpoint, which makes trials very, very long and complicated because these events occur uncommonly. Now, complete renal response is used as an endpoint, and studies of lupus nephritis can be accomplished in a much shorter time. Another example is polycystic kidney disease of the autosomal dominant form, where total kidney volume was qualified as a biomarker, as a prognostic biomarker. And then it's been applied in clinical trials, and it's now accepted as a surrogate endpoint for approval of new treatments. This greatly simplifies clinical development in that area instead of looking for progression to end-stage renal disease or doubling of serum creatinine. But biomarkers need to be used appropriately and properly validated. One of the things that's particularly important to think about is how the biomarker is going to be used in drug development. So this is a matter of determining what biomarker category it belongs to and how the biomarker will be used in a development program, because this determines what evidence is required. Is it going to be used for prognostic enrichment, going to be used for dose selection, or perhaps to support regulatory approval as a surrogate endpoint for accelerated or traditional approval? Looking at some of the literature in neonatology and speaking to my colleagues at FDA involved in the neonatology area, I could find a number of different applications of biomarkers in drug development in neonatology. So pharmacodynamic biomarkers can be important for choosing the right dose and demonstrating target engagement. Biomarkers can be used for patient selection or selecting patients for enriching clinical trials. And when you're talking about a potentially toxic therapy, biomarkers can be used to select patients at greater risk of progressing, where the benefit risk may be more justifiable for those patients and perhaps not for other patients. And biomarkers can be used to support efficacy assessment. And one important use is pediatric extrapolation. We had a public workshop on this a number of weeks ago, and some really excellent examples of use of biomarkers to extrapolate data from adults to children were presented. And then use as a surrogate endpoint is another potential use. But it's important to realize there are caveats for using biomarkers as surrogate endpoints. They're not a direct measure of the way a patient functions, feels, or survives. Instead, they're intended to reflect and predict the clinical benefit. So with the surrogate endpoint, the benefit risk is based on assumptions and predictions of benefits. And there are situations where biomarkers may fail to predict clinical benefit. For example, when they're not on the causal pathway of the pathogenesis of disease, they may correlate with clinical benefit, but not predict it. So when they're used in a new context, they may not work. Another example would be where safety issues come up that mean that clinical benefit that may be seen with the biomarker really doesn't demonstrate a favorable effect on the patient. So let me turn briefly to digital health technologies next. So mobile devices are being used more and more in drug development to collect patient health-related information. And there are a wide variety of possible uses of these new technologies. They can be used to measure adherence, to make sure that patients are actually taking or using the therapeutic agent. They can be used to construct novel clinical trial endpoints. It can be used for safety monitoring. It can be used for recruitment and retention. And in some cases, they can be used to allow remote clinical trials. So some of the data collection may take place in the patient's home. And patients who might not be able to participate in clinical trials because they can't travel to the study site can participate in the clinical trials anyway. So this can lead to more diverse patient populations and less burden for patients and their families. Here are some of the wide range of different sources of data in digital health technology. There are a variety of different biosensors. Smart pills are an example of digital health technology. Actigraphy is being widely used to measure patient function. And then there are a variety of interactive mobile applications that also fall into this digital health technology category. And I know you'll be hearing a lot more about that at this meeting. Some of the opportunities for digital health technologies in clinical studies are to study patient functioning, disease status, safety events, patient-reported outcomes. But there are challenges as well. It's important to make sure that the device is reliable and it's measuring what it purports to measure. There can be current concerns about missing data, making sure that it's measuring who and what it's supposed to be measuring. And then translating measurements into endpoints is not straightforward. It's not just a matter of putting a digital device on a patient and then looking at the outcome. You need to make sure that the data you get really corresponds to something meaningful for the patient. So when you collect data via digital health technologies, you need to collect the data, analyze it, and put it in the form of an endpoint that measures the way a patient functions, feels, or survives. And formulating the endpoint requires specifying a number of different things. What is being measured? So this may be steps, for example. What's the best time window for observation? Maybe it's 4 weeks. Would this be at the end of the study or at the beginning or over the full course of the study? And then what's the formula for translating the response into an endpoint? So this might be the change from week 1 to week 4 in the average daily step count. And I know these don't apply to neonatology, but of course there are similar applications in neonatology. But in the end, the question is the endpoint a clinically meaningful measurement of the drug effect, which it has to be to serve as an endpoint. So I'm going to turn next to real-world data and real-world evidence. So real-world data are data that are related to patients' health status and or delivery of health care that's routinely collected from a variety of sources. It can be electronic health records, it can be digital health technologies, and so on. So this is what the data are in real-world data. And then the evidence that comes from them is the clinical evidence about the usage and potential benefits and risks of a medical product that's derived from analyzing the real-world data. And the framework that FDA uses to evaluate the use of real-world data, real-world evidence, is to make sure that the real-world data are fit for use, to determine whether the trial or study design used to generate the real-world evidence can provide adequate scientific evidence to answer the regulatory question, and of course, to make sure that the study conduct meets FDA regulatory requirements. And I'm not going to speak further about real-world data, real-world evidence, because I know that John Concato is going to be speaking about this in more detail at a later session. So to end, I just want to talk about a couple of areas where innovation can be explored with FDA. One is the Clinical Path Innovation Meeting, and we've had a number of these on important topics, including a topic in neonatology, using an EEG-based measurement to assess pain in neonates. And I know that's really important because distress on the part of the neonate can reflect pain, but also other things. So that was a really interesting innovation. So at these critical path innovation meetings, we discuss the science, medicine, and regulatory aspects of innovation in drug development. Submitters can provide a request for a meeting, and then the FDA pulls together subject matter experts to consider it and provide feedback to the person requesting the meeting. And there's a guidance document that discusses this in more detail. We have a new pilot program in my office called iSTAND. It stands for Innovative Science and Technology Approaches for New Drugs, or iSTAND. It's a pilot program. We learned that there were some tools that didn't fit neatly into the category of clinical outcome assessments or biomarkers. And evaluating these tools needed to have a place in FDA. These tools include artificial intelligence-based tools and microphysiologic systems and 3D organoids to assess clinical pharmacology issues and so on. So this is a way for these tools that fall outside the usual framework to come and get attention to be qualified at FDA. And finally, I'd like to put in a plug for public-private partnerships like the Critical Path Institute. Qualifying biomarkers is highly resource intensive, and groups that are interested in qualifying biomarkers, like academic groups and smaller companies, may not have the funds or necessary data to qualify these biomarkers. And public-private partnerships like the Foundation for the NIH and the Critical Path Institute can play a really critical role. They can serve as an intermediary between patient groups, industry, academia, and regulators to develop novel drug development tools. They can collect trial data, integrate data sets, analyze and share data, and offer workshops to allow all the stakeholders to come together to share their views. So I just want to make people very aware that this is a really important mechanism to help develop drug development tools. And with that, I'll end. Thank you very much for your attention.
Thank you so much, Dr. Siegel, for your excellent presentation today. Really honored again that you were able to join us today. And I hope you would stay for the panel discussion and answer any questions coming in from the audience. Now, before I go further, I wanted to take a few minutes to honor and recognize a very special friend of International Neonatal Consortium who has been associated with INC since day one, since the day INC started, and has been our steadfast mentor and a friend throughout the 5 or 6 years of INC's existence. And that very special person is none other than Dr. Susan McCune, who was formerly the Director of Office of Pediatric Therapeutics within the Office of Commissioner at FDA. And she is now the Vice President of Pediatrics and Clinical Pharmacology at PPD. It was in part because of Dr. McCune's efforts many years ago that INC became a reality when she was able to convince FDA's NCPATH leadership that a public-private partnership focusing on neonatal population is needed. And that is how INC was born and INC started. Because of Dr. McCune's long-standing friendship, partnership, and mentorship to INC, we're honored to present our inaugural Friend of INC award to Dr. McCune. This award will be an annual occurrence, and we will continue this award every year in recognition of selected INC partners and contributors, and then we will also henceforth call it as the Susan McCune Friend of INC Award. So before I go further, I would like to invite Dr. McCune on the stage and ask her to offer a few remarks. Dr. McCune?
Kanwaljit, thank you so much for that kind introduction. I just, since you had the diagram, and I apologize because it's so big and it's so beautiful, I did want to let you know that the plaque did come and it looks really very lovely. I'm very humbled by this award. You know, this really takes everyone who is here at the meeting today to be able to accomplish all of the work that that we have done. I just wanted to take a couple of minutes to really thank the folks at the beginning. You know, we were talking about the establishment of public-private partnerships, and Dr. Siegel gave such a great lecture in terms of biomarkers and real-world evidence, real-world data, something that, you know, is critically important in the ink space these days. But this would not have happened without the vision of the folks that are on these two slides. The first being Martha Brumfield, who was open to the possibility of having an international neonatal consortium. And then the folks that were involved in the weekly meetings for a while and are my dear friends. And up front is Lynn Hudson, who was the scientific lead at the time for the International Neonatal Consortium. And then behind her, John Davis on the left, Ron Portman in the middle, and Mark Turner on the right. All folks that are well known to everyone in the International Neonatal Consortium. And I just want everyone, at least those folks, to recognize the title of my slide is The Starting Lineup, and that pays homage to the fact that for a very long time, we spent the first 5 or 10 minutes of every meeting trying to introduce Mark to American sports. And I will just give a shout out to John and the Red Sox right now, and I won't say any more because I'll jinx things. And just to let everyone know that the hockey season has started and the Capitals are looking quite good. So everyone's in trouble. But I just, all of the work for the International Neonatal Consortium would not have happened if it hadn't been for the folks that are on this slide. And then everyone has seen these in every slide deck that I have, but I just want to remind everyone, this is why we're here. This is all about collaboration. We all started in the sandbox very early on. The reason why we're here is because neonatal and adult diseases really don't overlap very much. But when it really comes down to it, this is really about the collaboration and bringing all of the stakeholders together in this space. And while I'm no longer a part of that government or regulatory stakeholders, I'm still a stakeholder in this process and very interested and invested in trying to make sure that we continue to move forward using, as Dr. Siegel talked about today, a lot of the data that are out there in terms of modeling and simulation, basic science research, clinical trials, and endpoints, as were so beautifully talked about in the previous talk. And then the IT delivery systems all coming together in terms of leveraging the insights from folks from academia, from government agencies, from industry, from patient advocacy groups, because at the end of the day, this is all about the impact to patients in terms of better dosing, more appropriate use of the current drugs, and increased access to new drugs, and ultimately improved neonatal drug labels. And so I borrowed this word cloud from the International Neonatal Consortium because I think it really speaks to the collaboration and why we're here. Collaboration is in the center of this, but really it's all about the parents, it's all about the families, it's all about our preemies and our babies, it's all about better care, and at the end of the day, making progress in terms of providing better medicines for neonates. So I want to thank Kamaljeet. I want to thank the International Neonatal Consortium. I want to thank all my friends. It's been a fabulous collaboration to this point, and it will continue to be just a leading group in terms of developing therapies for neonates. And I'm proud to have been part of that in the past, and I'm proud to be part of that continuing to move forward.
Thank you so much, Dr. McKeown. The honor is indeed all ours. And again, we are so proud and honored that we were able to provide you with this award today. And then, as I said, we will continue this award series in your name in the future from next year onwards.
Well, thank you. I'm very honored, but it makes me feel a bit old, but that's okay.
We will invite you to make the presentations next year. So I think
I will be honored and I will be honored and glad to do that. And thank you so much always for your support, Kamaljeet.
Thank you. And now on to the main discussion topics for this morning session, that is cell and gene therapies in the newborn population, in the neonatal population. So this session will have 3 lectures and a panel discussion. And this session is going to be chaired by Dr. Mark Turner. Dr. Mark Turner is the professor of neonatology and research delivery at University of Liverpool, and also chaired by Dr. Thomas Miller, who is the vice president and global head of pediatric development programs at Bayer Pharmaceuticals. And now I'm going to invite first Dr. Turner to provide brief remarks, and then Dr. Miller to provide brief remarks before we go on to the main presentations for the session. Mark, are you online?
Yeah, thank you very much, Ken Martin, and thank you very much to Susie for everything that she's done to support us. So we've organized this meeting because of the immense excitement surrounding cell and gene therapy for neonates. Cell therapies have great potential because the baby is a developing mass of cells. And if we can introduce some new cells in the right place, then we can harness that developmental capability to overcome some of the challenges that we meet on the neonatal unit. Genetic diseases are important. Each one causes an enormous burden on the families and also health services and communities, but collectively they're quite common. And so if we can find ways to address these conditions, particularly through platform technologies, then we're on to a winner. Now, while there are many neonatal diseases, some of which are directly genetic, like surfactant protein B deficiency, as we'll hear about later on, other diseases may be treatable in the newborn period. And so if we can treat people before they become unwell, then the newborn period, together with newborn screening, offers a great opportunity for truly preventive medicine that then cuts down the burdens and the costs. So while it's urgent and complex to develop these technologies, we do need to start thinking about what can we do to speed things up. What can the neonatal community through INC do to find out the best way to do these studies, the best way to prepare for these studies at site level, and the partnership with regulators and industry, putting families at the center of everything that we do. So what can we do to speed things up is a topic for this discussion. And we've got a number of enlightening speakers and then a panel discussion to think about that. And hopefully the end point of this will be some kind of agreement as to what INC can do, whether that's a working group that's working on consensus elements of a genetic or a cell therapy development program. We'll have to wait and see. But thanks to all the speakers and panelists for joining. I think Tom's going to talk us through some of the reasons why this is so engrossing for many people in the industry.
Thank you so much, Mark. Next, I'm going to ask Thomas Miller to provide some introductory remarks as well. Tom, are you online? Can you hear us?
Hopefully now.
Yes.
Yeah. Thank you, Mark and Kawaljeet. It's an honor to be here and to have the opportunity to open the session. I don't want to make anybody nervous, but we are going to start off with a pop quiz. So Adriana, if you wouldn't mind, please pulling up the notation. And a question for the audience that will poll in real time. How many interventional regenerative medicine clinical trials are currently underway around the globe? And by this, I specifically mean cell and gene-based therapies. A, less than 500, B, up to 5,000, C, up to 10,000, or D, greater than 10,000. So if you wouldn't mind clicking the poll and submitting your answer quickly, I promise we won't divulge any individual answer, but very curious to see the total perspective as you make your selection. Why don't we, for the sake of time, while folks are making their recorded answers, why don't we move to the next slide?
Tom, just go to the left side of the screen, click on the down arrow.
Thank you. There we go. So this was largely a stimulus for this meeting and the topic that we've selected for this year, because as Mark had indicated, there has been a very rapid expansion of interest in sponsored trials in this field. And we conducted a relatively low-tech analysis via clinicaltrials.gov. For those that might not be familiar with this resource, it's essentially a repository for all clinical trials conducted around the globe. And each country, including the US, has requirements with respect to clinical trial disclosures and updates. So it's a great resource to get a sense of how fields are evolving over time. And we started with an unfiltered assessment, essentially including any identifier or any citation. So if you look at the top left of your screen, you'll see total unique identifiers. We then moved down towards the next cut of the filter for trials that are interventional, those that are sponsored, those that enroll children, and those that allow enrollment of neonates, with an example that I provide. And we broke this into two categories: emerging stem cell-based therapies and emerging gene-based therapies. And for those of you that participated in the poll, if you look at the second row, the answer is not surprisingly a bit of a shock to the participants, I suspect. there are in fact more than 10,000 individual citations indicating that these are interventional based studies. When we look at the next cut down to sponsors, both modalities follow a relatively similar trend, but nonetheless quite a bit of activity across our industry partners, where almost every company that I'm aware of, certainly multinational companies, have declared some degree of interest in the field. But I think of most importance for our session today, if we look at the next two cuts down, those trials that allow enrollment of children and down to the neonatal period, it's clear that there's not only one or two specific programs, but quite a number of programs that have launched in recent years, and I suspect many more will follow. So I think a macro theme or question for this session, as we ponder the various challenges associated with these new modalities and drug development in children, specifically neonates, is what role, if any, should INC have in this conversation? And I'm very much looking forward to hearing perspectives from the various contributors. And before I leave and hand the mic back to Kanwaljit, some very practical examples with respect to emerging cell-based therapeutic programs. There's some great examples regarding human umbilical cord-derived mesenchymal stem cells, for example, for the treatment of bronchopulmonary dysplasia. We'll be hearing a little bit more about this topic as we go through our sessions later today. And we've had some fantastic breakthroughs as it relates to emerging gene-based therapies using viral vector-based delivery technologies to target monogenic disease targets. Most notably, successes in the fields of spinal muscle atrophy and Duchenne's muscular dystrophy. I suspect we'll see much more activity on both of these fronts in the coming years, and look forward to see what role INC will play in helping this field advance as expeditiously as possible. So with that, I'll hand the mic back to Kanwaljit to open up the session and speakers.
Thank you, Tom. So that was really illuminating indeed, that there are more than 10,000 clinical trials. I swear I didn't know that either, but it's good to know. That is why this topic is so important, right? So now we're going to start with the main sessions for main lectures for this session, and the first one up is results presentation for the GEMINI study, the genomic medicine for ill neonates and infants, that is going to be presented by Dr. Jill Marone, who is the pediatrician in chief at Women and Infants Hospital of Rhode Island. And without further ado, I'm going to invite Dr. Marone on the stage to present this result section.
Thank you so much, Kanwaljit. It's a pleasure to be here, and I'm happy to present the ongoing trial that myself and my colleague, Dr. Jonathan Davis, are PIs on. In order to develop therapies, we must first diagnose, and that will be the focus of the impact of integrating genomic testing into newborn medicine. We are heading a study known as GEMINI, Genomic Medicine for Ill Neonates and Infants. Oh, sorry. So we know there's an estimated 10 to 25% of neonates in our very NICUs that have an undiagnosed genetic disorder. And we are very also well aware that these neonates have a prolonged diagnostic odyssey. Under traditional genetic testing, this odyssey can last 5 to 6 years or longer. So there has been a push for more early diagnosis using emerging technologies into neonatal care. And how we integrate this technology really has been guided by lessons learned from prior ongoing genetic studies, the parent and caregiver perceptions of genomic testing at birth, these platforms that are emerging that you will hear more about as the meeting progresses, and ultimately healthcare economics. How valuable are these both to insurers and consumers, and what are insurers' willingness to pay? What we've learned from prior studies that were largely led by what is known as Insight, that was funded by NIH almost a decade ago, is that targeted use of these platforms may be better accepted by parents who have critically ill children rather than healthy infants. Studies have shown that Parents of sick babies in the NICU were more willing to participate rather than parents that had given birth to well, healthy babies. We've learned that rapid whole genomic sequencing provides a timely diagnosis of genetic diseases, but runs the risk of identifying incidental findings or adult onset diseases. This raises significant ethical considerations about widespread use. We also know through emerging economic assessments that the integration of these technologies could be drastically cost savings. An article put out in 2018 suggested that anywhere between $800,000 to $2 million could be saved by early integration of genomic testing. And that was really the basis for which GEMINI was born. It is an ongoing study that compares rapid whole genome sequencing to a commercially available targeted neonatal genetic platform known as Newborn DX. Our primary outcomes include time to confirm diagnosis on each of these platforms and what the economic impact of each is if integrated into neonatal care. For those of you unfamiliar, Newborn DX is a diagnostic panel. It is composed of 1,722 gene disorders known to affect neonates and children, and it's owned and operated by Quest Diagnostics. It is currently commercially available. It is not just simply part of a study, and any physician could actually order it through their channels. Newborn DX, though, is limited. Again, it only has 1,722 genes. And those genes were intentionally chosen to avoid some of the ethical dilemmas that we talked about, including adult onset identification of diseases. The platform cannot actually detect copy number variants, particularly if they're greater than 1,000 kilobase. So traditional platforms that we use, such as microarrays, are not able to be, the results cannot be seen with Newborn DX. So babies with microdeletions or duplications that are most commonly seen with our array comparative genomic hybridization arrays cannot be seen on Newborn DX. GEMINI is a prospective study with a goal of enrolling 400 neonates or infants less than 1 year of age suspected of having a genetic disorder. It's funded through NIH at the National Center for Advancing Translational Science. All subjects or probands and their parents, if available, undergo both platform testing. This is not a randomized trial. Everyone gets everything. Each laboratory performs its own phenotypically driven interpretation, and each lab is blinded to the other results. We will also determine diagnostic yields and economic utility of both approaches in the ill neonate. The sites participating across the country include Tufts Medical Center in Boston, Cincinnati Children's Hospital, the University of North Carolina in Chapel Hill, Mount Sinai in New York, the University of Pittsburgh, and Rady Children's Hospital in San Diego. Rapid whole genome sequencing occurs at the Rady Children's Institute of Genomic Medicine under the direction of Dr. Stephen Kingsmore. Newborn DX is performed at Quest Diagnostics in Marlborough, Mass. And for all findings, there is Sanger or PCR confirmation. Inclusion criteria include that you must be hospitalized. This is not an outpatient study. You have to be less than 1 year of age with a suspected undiagnosed genetic disorder. You cannot be born less than 23 weeks, have a major congenital infection, or a genetic diagnosis that fully explains phenotype. And this is interesting, and I'll talk about these results, but infants with an existing genetic diagnosis may be enrolled if that diagnosis cannot entirely explain phenotype. Potential subjects are identified by the clinical care team, and we use what's known as HPO or human phenotype ontology terms to classify the infant's symptoms, and that has to be also with the geneticist's input. For example, a cleft palate, a cardiac defect, microcephaly. You have the ability to request urgent testing as a clinical team if an infant meets the following criteria. These include that the baby's mechanically ventilated, has severe neurological complications such as unrelenting seizures, they're hemodynamically unstable, or simply at the request of the principal investigator or co-investigator at each site. If urgent testing is requested, this is an ultra-rapid sequencing and analysis, and the preliminary report is generated within 72 hours of specimen arrival at the lab. In terms of ethical considerations, parents must opt in to receive secondary findings approved by the ACMG for their infant and themselves. ACMG has just recently increased the number of these incidental findings related to genes to 73. Those are shared with the parents, and they have the option to opt in to know about themselves or their babies. These are not sought. They are simply incidental findings of the analysis. So all parents are told that if a negative report comes back, that does not mean they themselves or their child do not have these incidental findings. In terms of the childhood presentation, we will release the ACMG list only if they present in childhood and there's a specific treatment. So for example, BRCA is not released about the baby. Non-paternity is never revealed, and incest is reported to appropriate authorities for all enrolled mothers who are minors. Variants are classified based on the American College of Medical Genetics guidelines, and we only consider pathogenic, likely pathogenic, or variants of unknown significance. In terms of the variants are only reported if they are located on a gene that is causally related to a genetic disorder whose clinical factors clearly overlap with the proband's phenotype. In other words, we're not reporting every variant we see. We're only reporting variants that may explain the phenotype that's been presented. So where we stand as of today, I'm going to share with you the results of 374 neonates. I believe as of this morning, we are at 390. Of that targeted 400. This study was rapidly uptaken by both caregivers and families, and we will finish enrollment 18 months ahead of schedule. This chart reveals to you just the majorities of what we've seen. So for example, of the enrolled subjects, most are male, they are not Hispanic or Latino, and they are white. Most of these infants are actually born at term, greater than 37 weeks. And they're enrolled between 37 to 44 weeks, meaning that we enroll within the first 30 days of life. Time at diagnosis, also the majority are diagnosed within that neonatal period, ranging from 37 to less than 44 weeks of life. In terms of time to issue the report, here you'll see AthenaQuest and the Rady Institute. Median hours for Athena is 100, Rady is 120, and here are the means with the ranges. When we simply look at urgent cases, there were 92 infants that of the 374 I'm reporting that there was a request for urgent testing. Athena returns results with a mean of 89 hours and Rady 75. When we look at what we found, if we simply look at either it's pathogenic or likely pathogenic, 127 or 34% of those infants enrolled had that finding. A variant related to phenotype, an additional 17, and incidental findings were 3. 46% of our babies had no finding at all. What we're seeing in terms of these variants, sequence variants were 83% of the time, but copy number variants, those variants that the platform cannot detect, are being seen 16% of the time. And we have had one mitochondrial variant. When we look at results returned, we see that an infant with greater than 1 pathogenic or likely pathogenic diagnosis has occurred in 15 infants, or 4%. This was a little bit surprising to us, but it is true that we are seeing subsets of infants that have not just gotten one genetic disorder, they've had more than one. Infants diagnosed with a treatment that was immediately available, there were 42 or 12%, which allowed us immediate action. There were 49 infants, or 14% of families, that we did identify something that in the baby that extended to the family, and genetic counseling was recommended for the family. In 8 babies, there was withdrawal of life-sustaining treatment based on our findings. And in 3 of our infants, we were able to redirect care from simply comfort to curing them. To date, GEMINI has identified 173 novel variants that have never previously been reported in a neonate or infant. These are all actively being uploaded to what's known as ClinVar, which is the National Institutes of Health database for these novel findings. When we talk about concordance, what was unique about this study was that babies were undergoing 2 platforms simultaneously in their families. And when that happens, they may not always agree. 66% of the time they did. We had complete concordance from the reports we were receiving from Rady and Athena. But 31% of the time, there was actually discordant results that were getting called out, with partial concordance called out 3% of the time, and those had to do with babies that had more than 1 diagnosis. When you ask the question, well, why are they discordant? No surprise here, half of it was technical. And technical included, well, the gene actually wasn't on the platform. There were 1,722 genes only, whereas whole genome looks at every gene. So no surprise there. And again, because the platform cannot pick up micro deletions or micro duplications, they can't see those. And that was happening 16% of the time. But 37% of the time it was interpretation, and that was being done by the clinical care team, where they may see a variant, but decide it really isn't pathogenic or likely pathogenic. It's a variant of unknown significance, and that was not occurring infrequently. And then sometimes, again, depending on the platform and how many variants were being seen, you had a mixture of both. In terms of our clinical utility, all the caregivers are asked what they think about this platform. And what's interesting here is, yes, if a baby had a pathogenic or likely pathogenic variant called out, this was viewed as either very useful or useful 32% of the time. A VUS slightly less at 13% of the time. And if you only saw a secondary finding, it was only beneficial 2% of the time. But if we were able to report back to the physician, we don't see anything, they thought that was almost equally as valuable as if we did. And in total, 76% of clinicians found cumulatively, if we add them all up together, they thought this was either very useful or useful technology to have. Economic considerations, the families are asked to fill out monthly surveys inquiring about additional testing, medication, medical visits, hospitalization, and diagnoses. The medical bills accrued over the course of the first month of life and over the first year of life after discharge are collected and analyzed. And the economic impact of the testing of results on each platform is ongoing and will be made available when complete. In conclusion, targeted neonatal genomic sequencing in critically ill neonates and infants provides a diagnosis or identifies a VUS related to phenotype greater than half the time. The families and caregivers are provided with a clinically useful rapid diagnosis that directly impacts care and has been life-saving. But specifically designed neonatal panels, such as Newborn DX, currently do not have the capability to identify copy number variants, which in our study occurred in 16% of families. And the economic analysis are poised to identify which platform is the best to rapidly diagnose critically ill neonates. And with that, I just need to thank a few people. First and foremost, the families who participate. A huge shout out to Ann Kerfess, our GEMINI project manager. This study does not go on without her. Our GEMINI investigators listed here and the entire consortium that make this work possible, as well as the laboratory and staff at Athena Quest. Thank you.
Thank you so much, Jill. This has been fascinating, really illuminating results, and I'm pretty sure there are going to be questions coming in from the audience, and we'll get to those during the panel discussion. But for now, I will go right ahead to the next session, That is going to be a presentation that is going to be by Dr. Bernard Thibaud, who is a professor and neonatologist at Children's Hospital of Eastern Ontario. And Dr. Thibaud is going to be talking about challenges and opportunities for mesenchymal stromal cell therapies for complications of extreme preterm birth. So Dr. Thibaud, I'm going to give you the mic now.
Thank you very much for the introduction, and thank you very much for inviting me. I look forward to discussing with you these challenges, but also the promise of cell-based therapies for extreme preterm infants. Of course, the impetus is the challenge of protecting ever more immature organs, and we are in certain, to some extent, the victims of our own success because we have now pushed back the limits of viability almost to the limits of biological viability. If we take, for example, the lung as an organ, babies that are born around 23 weeks gestation are just at a lung developmental stage where airways and blood vessels get juxtaposed, so where gas exchange is actually feasible. Before that developmental stage, we may not be able to ventilate these lungs. So clearly, to make progress, we need some game-changing therapies, and regenerative medicine, and in particular cell-based therapies, have given some promise because these cells, because of their self-renewal, but more importantly, differentiation capabilities, these cells may be able to regenerate damaged organs. And the front runner in cell therapy over the years has become the so-called mesenchymal stromal cell, a cell that had been identified in the bone marrow as a hematopoietic stem cell supporting cell. These fibroblast-like cells were found to have some regenerative potential in experimental cardiac injury, and this led to a series of preclinical studies highlighting the potential regenerative potential of these cells. And so the International Society of Cell Therapy had to come out with minimal criteria to define these cells. And you can see that these criteria are multipotency, plastic adherence, and the presence or absence of certain cell surface markers. The multipotency is limited to the differentiation of these cells into bone, cartilage, or fat. And you immediately recognize the still rudimentary aspect of this definition. Nonetheless, numerous preclinical studies have suggested the therapeutic potential of these cells, including for complications of extreme prematurity. And research over the past 20 years have suggested that there's biological plausibility for using these mesenchymal stromal cells, or MSCs, for the prevention or treatment of bronchopulmonary dysplasia, as these cells exhibit pleiotropic effects that target multiple disease mechanisms involved in BPD. So not only would we have here a therapy that can prevent injury, but at the same time, the trophic function of these cells may also be able to promote lung development. And so this, these findings have spurred excitement in neonatology. Here I've summarized schematically 15 years of research showing that indeed mesenchymal stromal cells injected intratracheally or intravenously into experimental models of neonatal lung injury, but also other models mimicking sepsis, neck, or brain injury, for which we don't have currently specific therapies, that these cells seem to be organ protective. The other important piece of information is that contrary to the original belief that these cells would integrate and replace dead cells and regenerate organs, these mesenchymal stromal cells do not engraft, but they seem to act via bystander effect, releasing bioactive molecules or interacting through cell-to-cell contact to exert their therapeutic benefit. And this has been shown by injecting either the conditioned media or so-called extracellular vesicles that are shed by these mesenchymal stromal cells. And these conditioned media or extracellular vesicles have the same effect as these mesenchymal stromal cells. And so there is understandably great excitement for testing mesenchymal stromal cells or their derivatives in clinical trials. But we also know that the clinical translation of these disruptive therapies is unacceptably slow and often fails. And to this effect, we developed an approach, an evidence-based approach to ensure the timely and safe but also successful translation of novel discoveries. And I show this slide for two reasons. One, to show that it requires a multidisciplinary team that includes, for example, a health psychologist or health economist, and also to highlight the artistic capabilities of my then 16-year-old daughter, who designed the logo for the incubator concept. That's how we call the approach, and it's obviously an acronym. And you can see here how the incubator concept inserts itself in the classical pathway that leads from discovery to preclinical exploratory or confirmatory studies to then lead to clinical trials. And in between, we have inserted this incubator concept, and I will show you 2 or 3 aspects of this approach. And you can see that at each step, whether it's between the preclinical processes or between preclinical and clinical, the arrows point in both directions, suggesting that at some point we may have to go back to the drawing board and assess new questions that arise as we move from one part, stage 2, stage 3, or preclinical to clinical. And so here we did a systematic review and meta-analysis, not of clinical trials, but preclinical MSC studies in experimental models of BPD. And this was an interesting exercise where we screened over 1,000 data. In the end, at this point, we had 25 studies that were included in the analysis. Here, this slide shows the effect of mesenchymal stromal cells on lung structure. And you can see, except for 3 studies, all suggested MSCs had a beneficial effect in experimental BPD. I will skip over the next 3 slides because we're a little bit behind. They only show a classic experiment using the rodent model of BPD. The impressive effect of MSCs on lung structural prevention, but also safety in 6-month-old rats. However, when we look at the risk of bias in these systematic reviews, we can see that there are major shortcomings in these preclinical studies, mostly in terms of reporting the data and randomization. And now we have available guidelines, such as the ARRIVE guidelines, that have as a goal to increase the rigor of preclinical studies, since they are the prerequisite to lead to clinical trials that are time-consuming and expensive. Another interesting aspect was that all studies that were included were only performed in neonatal rodents. No large animal studies had been performed. And here we had the privilege to work with the only facility that is capable of delivering premature non-human primates and ventilating them for 2 weeks, exactly the same way we would treat our preterm babies in the NICU. And this setup provided an ideal platform to study these mesenchymal stromal cells and establish feasibility, safety, and efficacy of these cells to obtain regulatory approval and move to a phase 1 clinical trial. And these cells were injected intravenously right at birth, so we could study the immediate effect of MSCs and eliminate any infusional toxicity. And over the two-week study period, we could monitor some other safety and efficacy data. And of course, because it's animal studies, we have the luxury of then taking out the lungs and exploring them histologically. Here, I just show you one piece of data using forensic analysis to detect human mesenchymal stromal cells in the body of these non-human primates. And we were incapable of detecting any cells confirming the absence of engraftment of these cells, adding additional safety data. Of course, one of the main limitations or reasons of failure of clinical trials is lack of enrollment. And here we ask parents and clinical colleagues what they thought about giving stem cells to babies to detect obstacles and challenges for such a clinical trial, but also opportunities. And so we interviewed parents that were that had babies, preterm babies hospitalized at the specific age where we would enroll them into a potential trial. And we also interviewed colleagues across Canada and asked them specific questions to identify these barriers and opportunities for a clinical trial. Some of the answers are obvious, but others were very insightful. And the most important, I think, piece of action that emanated from these studies was the development of an animated informational video for parents that we thought would provide them with additional confidence to participate in a clinical trial, as this video would answer some of their questions. So here, I'm not sure how I can activate this video.
We will play it right now.
Okay, so I will just play, thank you. I will just play like 30 seconds of the video to give you the impression of what that video looks like. Please go ahead. Health Canada. Okay, can we move to the next slide, please? Thank you very much. So this video was very well received by parents, and we tested it in the observational arm of our phase 1 trial. Here again, it's the HULK-1, Helping Underdeveloped Lungs with Cells, a phase 1 safety and feasibility study. You can see again the multidisciplinary team that it takes to launch such an endeavor. And here is the experimental design, where we decided to start with an observational cohort to make sure that we had all our ducks aligned and that the experimental design was appropriate. The patients were eligible if they were born less than 28 weeks, intubated, mechanically ventilated. Initially, our FiO2 requirements were above 35%, but the observational cohort showed us that we could enroll babies that were on any type of FiO2, providing already one beneficial aspect of this observational cohort. And babies had to be between 7 and 20 days of life. And this is mirrors a little bit the babies that would typically be considered for postnatal dexamethasone if they are, as we say, stuck on the ventilator. We are also taking some blood and tracheal aspirates as surrogate markers, record, of course, clinical physiological adverse events, and then evaluate this informational video for parents. We completed this observational cohort 3 weeks ago. It was, of course, delayed because of COVID, but we're now ready to embark into the interventional cohort. It will be a dose escalation study to determine the maximum feasible dose for a phase 2 trial if this phase 1 trial goes well. And so we look forward to starting this study by the first quarter of 2022. Here are the endpoints, all focused on, of course, safety and feasibility. So despite the excitement in the neonatal field and other fields as well, I would like to highlight that cell-based therapy is still in its infancy. And I like the analogy with the cell phone because you can see that from the development of the first rudimentary cell phone in the 80s to the appearance of the first iPhone in 2007, over 20 years have passed. And this is just technology. Here, when we talk about cell therapy, we're talking about the biotechnology, and we can imagine how long it will take us to figure out all the details. And here again, as an illustrated example, in cell manufacturing, the product is actually the process, similar to making a cell phone. And here, this diagram illustrates very nicely the complexity of making a cell product and the numerous parameters that can affect cell function and, as a consequence, the results of any clinical study. Here, there are some particular aspects. For example, over time, we'll be able to genetically modify a given therapeutic cell. We also will have to answer the question whether we should freeze the cells or use a fresh product, whether the cells after they were thawed, we should culture them or inject them right after having thawed them, which is logistically more feasible. Likewise, should we provide the cells or should we provide what the cells are producing, such as extracellular vesicles? And there's a trial, a phase 1 trial currently going on, testing the safety of these extracellular vesicles. Finally, there is a great controversy around these mesenchymal stromal cells. I mentioned originally the still rudimentary definition of these cells. And in a scoping review that we recently finished, we showed that mesenchymal stromal cells, whether it's in preclinical or clinical trials, are inconsistently described, 18% only of the literature. There's inconsistent reporting of mesenchymal stromal cell characteristics and culture condition, and I just showed you how complex the manufacturing process can be. And there's a lack of completeness in reporting clinical studies. And here we have now added this aspect to our incubator concept. We'll develop a Delphi method to better define mesenchymal stromal cells and establish clinical reporting guidelines to instill rigor and enhance the success of the potential clinical translation of these cells. And so in conclusion, I think there's biological plausibility and a strong preclinical rationale for testing these cells in clinical trials. These trials will enhance our understanding on the safety, the timing, the dosing, the route of administration of these cells. But in parallel, obviously, much more needs to be learned about the biology of these cells. We have to fine-tune the manufacturing process. And you can see that in parallel, improvements in technology will facilitate and optimize these manufacturing process, providing us with better quality cells. We hope that the incubator will unravel its potential impact in accelerating the safe translation of these discoveries. We have a few in store here. I would like to thank all the teams, the preclinical team that worked on the HULC on mesenchymal stromal cells, providing the preclinical rationale. We still are working on endothelial progenitor cells for other indication, but also on some interesting adeno-associated viral vector gene therapies for surfactant protein B, where we have very interesting preclinical data and are trying now to manufacture a gene therapy for clinical trials. With this, I would like to thank you for your attention and hope to answer all your questions. Thank you very much.
Thank you so much, Dr. Thibaud. So this was really enlightening, and I especially like your analogy to of advancement in cell and gene therapies to the advancement in the cellular telephone technology. Yeah, pretty fascinating indeed. Thank you. I'm pretty sure there are going to be questions from the audience, and during the panel discussion, I will read them aloud to you so you can answer the questions. Thank you so much.
Thank you.
Thank you. And next up for this session is a presentation by Nicole Lamarca, who is the Global Medical Director and Therapeutic Spinal Muscular Atrophy expert at Novartis Gene Therapies. She's going to talk about spinal muscular atrophy. Early diagnosis and intervention is crucial. Nicole, so I'm going to give you the mic now.
Great, thank you all so much. So I'm going to try to go quickly so we have time for our presentation from the panel coming up. But thank you again for having me. My name is Nicole Lamarca. I'm a Global Medical Director at Novartis Gene Therapies working on Zolgensma. But just to give some context for my presentation today, prior to joining, my career has been spent in pediatric neuromuscular disease at Columbia University. I had the wonderful experience of being able to work prior to any disease-modifying therapies for SMA. So working on the natural history studies, then really embarking on the drug trials. So for the 3 drugs that we have right now for SMA, being able to participate as a co-investigator for those. And then allowing us to then really start our newborn screening for SMA. So working on those pilot studies in New York State, getting those approved, developing a program at the institution, and then once we had approved therapies, getting those to patients. So I hope to speak to some of my experience today, and kind of how that can help with other diseases for neonatal patients as that moves forward. And I'm sorry, I did not advance the slide. I'm not sure. Did that work? So.
Nicole, try the up and down arrows on the left-hand side. Yes.
Oh, how's that? Okay, great. All right, so in this session, we're just going to kind of quickly review SMA and the burden of disease that patients have, highlight the importance of early diagnosis and intervention, present some of the latest developments in newborn screening for SMA worldwide, and discuss the experience in treating neonates in clinical trials and kind of how to handle that, especially for those neonates that are still pre-symptomatic. So as many of you know, SMA is a rare neurodegenerative disease caused by the biallelic deletion or mutation in the essential SMN1 gene. SMA is characterized by an irreversible loss of motor neurons, resulting in progressive muscle weakness. So once those motor neurons are gone, they cannot regenerate, they don't come back, despite the disease-modifying therapies we have on the market right now. The incidence of SMA is approximately 1 in 10,000 to 20,000 live births worldwide. And again, carrier testing can be done. And then we'll talk a little bit about newborn screening as well. Patients with SMA present across a gradient of varying phenotypes. A lot of that is dependent on the number of copy numbers they have, so it correlates with their disease severity. But again, it's not necessarily predictive, but does correlate. And SMA, I think, is probably the most important thing I'm going to stress here, is that it's a very complex patient journey. Regardless of the disease-modifying therapy chosen, there still requires this huge multidisciplinary approach and treatment management for these patients. We know that early diagnosis is essential for improving outcomes for patients with SMA, especially for those with the most severe forms of the disease. So again, SMA is caused by this biallelic deletion or mutation of the essential gene called the SMN1 gene, resulting in deficient levels of the protein. Patients rely on that SMN1 gene. However, there is an SMN2 gene, the survival motor neuron 2 gene, that is working, but it only produces about 10% of functional good length protein. So without sufficient SMN protein, patients with the most severe forms of SMA can experience irreversible motor neuron loss, have progressive physical disability, and experience premature death. This is where the different disease-modifying therapies come in. They work on different aspects. A product like Novartis, so that's Zolgensma, works on the SMN1 gene, replacing the SMN1. Okay, so now we can look a little bit about the natural history of the disease. And I just bring this up because it really stresses the need for early rapid intervention. In SMA type 1, patients, natural history shows, and this is through PNCR network, as well as through the Neonates trials that were done, that over 90% of untreated patients with SMA did not survive or required permanent ventilatory and nutritional support by the time they reached their second birthday. So by 2 years of age, they required either invasive ventilation or invasive feeding in order to sustain themselves. Again, I think important to note that this irreversible loss of motor neurons begins early in the perinatal period. So especially those with fewer copies of the SMN2 gene, we know that even in utero, patients are being affected. So rapid treatment is required, and in order to see best effects for these children and best outcomes. Again, early therapeutic intervention is critical to halt the loss of those motor neurons. And we know that lower motor neuron degeneration starts early in life and cannot be reversed. This graph here that you're looking at shows the loss of motor neurons in patients with SMA type 1, type 2, and type 3 if left untreated up until the age of 5 years. Type 1, again, being the most severe, they can lose as many of 95% of their motor neurons by 6 months of age. Clinical diagnosis may not happen until that child is 6 months of age. They may just be viewed as a hypotonic, lazy baby. So that is why it's imperative to really screen these babies with newborn screening so they can be picked up early and treated early. I'll talk a little bit about SMA carrier testing and newborn screening. So these are being implemented in several countries across the globe. In the US, SMA was included in the recommended uniform screening panel on July 8th, 2018. So almost 2 years after the first FDA-approved therapy for SMA. SMA carrier testing and newborn screening are now being implemented outside of the US as well, throughout Europe and Asia Pacific, Latin America as well. At least 13 countries are starting to implement either carrier or newborn screening testing worldwide. The number of newborns screened for SMA is expected to steadily increase over time, which is an exciting prospect for these children. As we can see here in the US, we may have to click through the slide to get it to populate. Let's see. So you can see here which states are screening for SMA. So newborn screening is a public health initiative that is performed across the United States. This test, as many of you know, is performed within the first 24 to 48 hours after birth for a variety of congenital disorders. SMA is now one of them. I think it's incredibly important to note that really SMA getting to the RUSP had a lot to do with the support from the patient advocacy organizations, especially Cure SMA really rallied to support this initiative. So we're thankful for that. There were many pilot programs that happened as well, and really showing the feasibility that it could be done has been tremendous for these patients. But as you can see, there are some states that are still not screening. It has not gotten started in the States. I know COVID caused some of those delays to happen. So it's important, again, that these babies are being picked up clinically as well as through newborn screening. So here I'd like to talk a little bit about how early diagnosis and treatment has led to improved outcomes for patients with pre-symptomatic SMA. And I think that's probably where we will have the biggest impact for these patients, is intervening before those motor neurons have died. So there are 3 clinical trials here that you can see. So SPRINT was done by Avexis, which is now Novartis Gene Therapies, Nurture through Biogen, and Rainbow Fish through Roche. And these are all pre-symptomatic studies. I guess what I really wanted to bring to you today is just the experience that we had kind of embarking on these 3 projects, and some of the positives, but also challenges that came with trying to enroll patients who were pre-symptomatic, so to their families, no symptoms of SMA could be seen at all. And really how that affected enrollment in clinical trials as well as treatment later on. So many of these children, especially early on, were identified either through prenatal genetic testing, so carrier testing that may have been done. There may have been a family history of SMA, so they were prepared to test the child, or it could have been newborn screening initiatives. As time has gone on, it's been less of the pilot studies, but more part of the RUSP panel for enrollment in these trials and whatnot. Through these studies, we've seen that early treatment for pre-symptomatic patients for SMA has shown improvement in survival, ventilatory support, feeding support, and motor outcomes, which is really what we want to see for these patients. Data we have, again, across all 3 companies, all 3 drugs have shown that implementation of newborn screenings will facilitate treatment for these babies pre-symptomatically, maximizing their clinical outcomes. Enrollment for these patients was often difficult. Many families were not familiar with SMA. Again, these children were healthy appearing newborns, And there were also lots of questions surrounding prognosis, right? So we talked about how you can correlate with their SMN2 copy number, how severe their disease will be, but again, not prognostic. And as time went on and there were disease-modifying therapies were in various stages of approval, there were always lots of questions about different conversations with the families, but really it was imperative to have a multidisciplinary team. And I'm glad, I think all three talks today have talked about the multidisciplinary team working with families. So supporting the entire family during the clinical trial. It's not just the baby and the parents, it's the entire family often, as well as potentially leaders in their community. So trying to get everyone on board is critical. So preparing for that when you're developing the clinical trial. So being aware that you'll be having conversations with extended families, being prepared to support postpartum parents. So for these clinical trials in SMA, the babies were treated before 6 weeks of age. So really trying to make sure that you're sensitive to the needs of the family at that time and working as fast as possible in a very coordinated, seamless team. And making sure you're speaking with one voice, I think, was key as well. So even if you're, as a team, saying the same thing, if it's said slightly differently, that can cause a lot of confusion for families. So, and then potentially delays in therapy. So after the enrollment phase, even going on to maintaining them. So that's another thing. So maintenance in the clinical trials. Some of the products are one time. So Zolgensma, the product, the gene therapy from Novartis Gene Therapies, is one time. So you're dosed once and then you continue on in the clinical trial. A product like nusinersen is, after loading phase, is dosed every several months, and risdiplam for Rainbow Fish with Roche, that would be given daily. So again, maintaining them in the trial is key. Working to support the entire family as they participate in the trial, making sure transportation and childcare is covered for them, really to try to get continued support and involvement, and making sure you're really focused on the patient. I also want to take this opportunity to stress that although they're in clinical trials, making sure these patients are receiving standard of care as well, so that they're continuing to meet with their multidisciplinary care teams as needed, is key for success for these patients. Right, so my final slide here, and I tried to go quickly, but I hope you have questions during the question and answer session. It's just overall lessons learned from SMA, and I hope that this can help to guide some of the other programs that may be going on in the future for neonatal research trials, but really stressing that newborn screening is crucial to ensure early diagnosis of genetic disorders, to allow for that prompt treatment or initiation in clinical trials. It's accepted internationally as a preventative public health program, and it's critical for early identification of disease. It allows for the identification of disease before symptom onset, eliminating any sort of diagnostic odyssey that these patients typically would have gone through, and really allows you to intervene before disease progression can occur. And this early detection, diagnosis, and treatment through newborn screening can really significantly reduce morbidity and mortality, and may facilitate adequate genetic counseling and the proper education to families about available opportunities for them. So I thank you all so much, and I will turn it back over.
Thank you so much, Nicole. This was really wonderful talk, and I greatly appreciate your time today and talking about your experience with SMA and all the various different clinical trials and what's the best way to actually maintain the patients onto the clinical trials. Really very crucial. Thank you. I'm sure there's going to be questions coming in from the audience, and we'll get to them in a short order. But right now, we are going to jump right into the panel discussion. We have a set of esteemed panelists that we have gathered here for our session, and I'm actually going to ask each of them to come and give a very brief perspective. And the panelists that we currently have online are Dr. Elizabeth Hart, who's the branch chief general medicine at the Centers for Biological Evaluation and Research at FDA, Catherine Walsh Rockley, who's a genetic counselor at University of Pittsburgh Medical Center. We have Dr. Lauren Servais, who's the professor of pediatric neuromuscular diseases at Oxford University. Then we have Dr. Deborah Gill, who's the professor of gene medicine, also at Oxford University. Then Dr. Richard Scott. I think Richard Scott is actually not present today. He had the last-minute conflicts and he was unable to join the call. But we do have, for a parent perspective, we have Aaron Ward, who is the co-founder and president at the MTMCNM Family Connection. I'm actually going to individually invite each of them, provide brief remarks and perspective, and then I'm going to go on to asking the panelists questions. So first up is Elizabeth Hart. Dr. Hart, if you are online, I'm going to invite you to provide some remarks.
So good morning, everyone. Thank you for including me. As you've heard, I am the branch chief of General Medicine 1 in the Office of Tissues and Advanced Therapies in CBER at the FDA. And I just want to emphasize how much we are interested in really partnering with the community to advance therapies for children, infants, neonates, etc. We see this as an area of great need. And as you've heard, cell and gene therapy is an area where there has been a lot of new research and development. And we've had the number of new investigational drug applications increase dramatically over the past few years, and we anticipate that will continue. And most of what we're seeing, especially in gene therapy, is in the area of rare disease. Almost 3/4 of our applications are for rare diseases, and a lot of those include children and infants. And so we're really interested in working with you to advance these therapies. And we have regulations that we absolutely need to follow, but there are also opportunities for flexibility. And so we look forward to working with you. Thank you.
Thank you so much, Elizabeth. In case any questions come in from the audience, I'm going to be directing to you after we're done with this panel discussion. Yes, thank you so much. And I'm pretty sure, you know, all of our INC's industry and academic members are also very eager to work with CBER on you know, on a mutual basis, basically leading to advancement and cell and gene therapies in newborns. And we at INC also look forward to working with you on a much closer basis on this. Next up is Catherine Walsh Rockley, who's the genetic counselor at the Division of Genetic and Genomic Medicine at University of Pittsburgh. Catherine, I'm going to ask you to come online and provide brief remarks as well.
Okay, can you hear me?
Mm-hmm. Yep.
All right, great. First of all, thank you for including me in this conversation. It's fascinating, and I am involved in research here at Children's, worked with Jill and colleagues on the GEMINI project as well. I am so pleased to see the work that's being done in terms of informed consent for with the families, the involvement of the families in contributing information to how trials might be done and how the consent process can go forward. One of the things I was planning to talk about was the use of apps and videos as a way to educate families. And it's nice to see that Dr. Thibault has been doing that with his program. I think as the number of families who are eligible to participate in these trials increases, we're going to have to get creative. We're going to hear later this afternoon or this evening about newborn diagnosis. And as our first speaker today said, while these conditions are individually rare as a group, the numbers are growing in terms of those that are eligible for intervention. I almost wonder, and I know there are some initiatives happening in the US with this, whether we really need to be looking at overall genetic literacy in the population, because essentially anyone could become a candidate for a gene or cell therapy. So are there initiatives that the organization can support in terms of educating the community at large, so that as we get to an informed consent process, the time and effort it takes the involved professionals is less. There are also lots of ethical issues that are my area of interest, but I know we have an ethicist speaking later in the program as well. So I'll defer on those, but happy to answer any questions. Thanks.
Thank you so much, Kate. Glad to have you here, and I'm sure there are going to be questions, and I'll be referring them on to you. Next one up is Dr. Laurent Servais, who's the professor of pediatric neuromuscular diseases at Oxford University, UK. Laurent, if you are online, may I invite you to provide brief remarks?
Sure, and thank you very much for inviting me to this very exciting discussion. So indeed, I'm Professor of Pediatric Neuromuscular Disease at the University of Oxford. I have a typical British accent, as you can notice. I'm basically from Belgium, and I've had the chance or the malediction, I don't know, to be first line in the story of spinal muscular atrophy treatments over the last 5 years. And it has been a fantastic but exhausting time, where we have learned a lot about what could be a success in the rare disease field. And one of the things we've learned, which is very relevant today, is how important it is in the context of a neurodegenerative disorder to treat early, and probably to treat even before the symptoms, right? So in SMA, all over the world now, there are several newborn screening programs. It's quite an easy case, SMA. It's always the same mutation, right? Very easy to do by PCR. It's not exactly the same for all rare diseases. So my strong conviction is that we need to move forward to new techniques of newborn screening if we want to be ready to accommodate the number of clinical trials, the number of development, and in the different rare diseases. Because obviously in several diseases, we know already that earlier is better, and probably sometimes even only before the symptoms is efficient.
Thank you so much, Dr. Servais. Then next up, we have Deborah Gill, who is also from Oxford University, and she's a professor of gene medicine there. Deborah, do you have any thoughts, feedbacks, comments?
Yes, I'm based in the UK, as you say, and I'm interested in doing gene therapy for neonatal lung diseases. And my background is in the development of a platform delivery system for genes to the lung, and we've been doing that in adults for treatment of cystic fibrosis. And we want to take all the lessons that we've learned for like manufacturing and toxicology, and now apply those to delivery of genes for neonatal gene therapy, in particular surfactant deficiency, and in particular for delivery of the genes for surfactant B and ABCA lipid transporter. And aside from the generic challenges that we all kind of talk about in all of our work with neonates, there are some specific challenges which we have to think about. So these babies present at birth with severe surfactant deficiency, and they need a rapid screen while they're on the ventilator. And the challenge for us is to identify the extremely rare patients. So something like 1 in a million has been quoted for surfactant protein B. And in order to be able to do this, we really need an efficient and informed clinical network. And that's kind of what we're beginning to, that's why I'm here, and why we want to make connections with people across the world.
Thank you so much, Deborah. Great comments. And then next up, we have Erin Ward to provide a perspective from the side of the patient and the parents. And then Erin Ward is the co-founder and president of MTM CNM Family Connection. Erin, if you are online and you can hear me, I would invite you to provide brief remarks as well.
Thank you so much for having me and for having patient and family representation here today. Also, as the mom of a NICU graduate, this is really near and dear to my heart, and thanks for all the work that you do, not only in the evident today, but every day. Also, as president of MTM CNM Family Connection, I work specifically with the myotubular and centronuclear myopathy communities, and many of you may know that we have our first clinical trial that there's now been 4 unfortunate deaths connected to. So this is very much work that I'm doing every day with the family population, but also working closely with biotechnology partners. And additionally, I have some experience with 15 years working in a medical ethic program specific around NICU conversations and working specifically with multidisciplinary teams and how to have difficult conversations. And it's actually been very much on my mind how the NICU community might be really well positioned to help all medical teams be thinking about how to have these really high stake conversations that really often surround, in many cases, could be life and death. Some just very general remarks. I think I very much, having lived in the NICU for many months, know that things can happen very quickly for a patient and family. So really, it's about what Nicole said: balancing the current needs and care of the infant, at the same time looking ahead to what potential trials might be possible. I think that's something that's really been coming to light for our community. We just recently had a patient focused drug development meeting with the FDA and talking about risk and benefits. And I think it's really important for the broader community to really think about how just high mortality rates do not necessarily have a direct correlation in how people feel about the risk that they want to take. We can't have those types of conversations being made in a really isolated way without patient and family involvement, because oftentimes what those decisions are about is really about a family's way that they define quality of life. So both on an individual level for families that are talking about being in a trial, having a team to really surround them around those important conversations, but also in a bigger way, having patient communities be involved in the process from the very beginning, talking about meaningful outcomes, talking about how to be part of the entire drug development process. So those are just some general comments, but this is really important work, and I thank you all.
Thank you so much, Erin. Your comments about having family and parent organizations and parent representatives involved at the very, very beginning of the process is extremely important, and we all agree with you on this. This has been a great discussion today. I do have several questions that I've lined up for all of the panelists, and including some of the people who actually spoke, had main presentations today. What I'm going to do is read the questions aloud. Some of those questions have been directed to specific individuals, and if you can just come online and answer the questions, that would be great. The first question that I've gotten is for Dr. Jeff Siegel, who was a plenary speaker today. I had a question that could you please elaborate on the notion of using quantitative disease models to provide the underlying evidence for quantification of biomarkers?
So this is a really interesting area. We think that modeling can be quite insightful to understand how to connect biomarkers to clinical outcomes. Sometimes it requires pulling several different data sets together. Keeping track of how to integrate different data sets to provide models that provide more precise quantitation is helpful. But the ability to include covariates in the model to be able to provide confidence intervals that are able to accurately predict outcomes is really helpful. So I think this is an important area and would fully support further work in this area. An example just real quick is cystic kidney disease. Modeling was done with the data on total kidney volume and provided precise confidence intervals on how total kidney volume related to renal dysfunction, which was very helpful in qualifying the biomarker.
Thank you. Thanks, Dr. Siegel. And I'm pretty sure some of these approaches can very well be adopted to some of the neonatal conditions as well, which we fully do intend to, and work with your office on some of these. And there was another follow-up question also directed to you. What is your take on interconnectivity between digital health technologies, artificial intelligence approaches to optimize signal processing, and pharmacometrics approaches, and how do you link everything up together?
Yeah, another really interesting question. Analyzing these vast amounts of data that we get is a challenge, and we can develop our own algorithms for analyzing them and interconnecting them. But artificial intelligence often provides insights that scientists may not see initially. So we think there's a lot of value in applying artificial intelligence, machine learning techniques to analyzing data, including digital health technology data. When you're developing a biomarker, however, it's important to lock down the AI algorithm that's being used. That shouldn't vary continuously. So do your AI, lock down the algorithm, and show the algorithm that analyzes the data. Subsequently, if you're going to modify the algorithm using new AI analyses, you want to do that in an iterative process, showing the validity of the new method and how it's superior to the old one. But I think this is a great approach, and certainly one that's being used more and more.
Thank you. Thanks, Dr. Siegel. And then, then there are a couple of questions. One was a question that was directed to both Nicole and to Laurent. Both of you have experience with spinal muscular atrophy and Duchenne muscular dystrophy gene therapy studies. And beyond study results, what has been your biggest learning from these gene therapy development programs? Maybe first I'll ask Laurent to respond to this question.
Yes, sure. I think that the relationship with time is completely different for spinal muscular atrophy and Duchenne. And the second aspect is that the understanding of safety is completely different. Let me explain. When you have SMA type 1, perception of safety, I mean, you are, as a parent, ready to take quite a significant risk, right? Because you understand that this is a very severe condition and that, you can accept a certain level of risk. For a 5-year-old with Duchenne doing quite well, it's more difficult. And if you speak with a teenager with Duchenne, he will not be happy to make major risk. So perception of risk is different, but perception of time is different, because in SMA, there is no doubt that the earlier is better, right? But in Duchenne, it's not completely clear, because we know that our muscle mass will increase with time. So if you give a dose for patients with 4 kilo, it means that when these very patients will be 60 kilo, there will be a dilution, right, in a muscle mass that is 15 times bigger. But on the contrary, if you wait too long, at that time, there is no muscle anymore, and there is no benefit to expect. So which means that between these two extremes, there is somewhere a sweet spot, and nobody knows when is this sweet spot. The second point is that the sweet spot is probably different from one to another patient. Patients with a more severe phenotype have likely a sweet spot earlier on, and patients with a less severe phenotype later on. And of course, if a treatment is available other than gene therapy, it could change the time of the best window of opportunity for gene therapy injection. So I think that Duchenne as an extra layer of complexity in terms of timing of injection in comparison with spinal muscular atrophy. So that's two basic learnings of both experience.
Alrighty, thank you. Thank you, Lauren. And then I think this was a similar question that was also referred to Nicole. Nicole, do you have any thoughts on this as well as with your industry hat on?
Sure. So I think that industry hat and then also clinician hat as well is the need to continue multidisciplinary care for all of these patients. So whether it's Duchenne or SMA or any condition that despite the therapy that they're continuing to receive standard of care despite being in a clinical trial, I think has been a huge learning for all of us, and I think one to remember and keep in the back of our mind as well.
Thank you, Nicole. Again, even though I'm referring these questions to individual panelists, if anybody else within the entire panel has any follow-up thoughts on some of these questions, please feel free to raise your hand and answer the question. And the idea is that we would like to simulate like a natural discussion with everybody. This is a question to Aaron regarding informed consent. How do regenerative medicine, cell and gene therapy studies differ from other type of programs, specifically, related to assurance of caretaker and parent understanding of the trials?
Yeah, I like Kate. I'm very interested in informed consent, and especially looking specifically at gene therapy and what we understand at this time, that potentially one dose is really where you're looking at. We're hopeful that in the future there'll be development of redosing if you don't get the dose right, but that weight of making a decision and an idea that it's a one and done therapy at this given time, I think there is a greater elevation of and need for clarity and full understanding for informed consent. And kind of similarly, in conditions like SMA and tubular myopathy, where in the infancy and the NICU, things can change on a day-to-day basis with an individual's condition. And I can imagine that there could be a situation where, at one point in time you have a conversation with your family and they're wanting to move forward with gene therapy and something happens that excludes them now from the trial, that these conversations have to be just all evolving, not only for the individual families and patients, but also, my hope is that we can get to a place where when these high stake dosings occur, that if there are any evidence of serious adverse events or anything that it's an iterative process where informed consent continues to evolve based on that knowledge that's fed back into the system in a very timely way, which is a struggle.
Right. Thank you, Erin. Yes, Elizabeth, you had a question. You had a follow-up, right?
So I just wanted to add on to that we do indeed recognize that there are a lot of potential long-term risks associated with these therapies, and along those lines, we've created a guidance on long-term monitoring related to gene therapies for investigators, and those are things that should go into the monitoring of the clinical trial and the long-term follow-up, and are also important to be in the informed consent so that participants really can judge the risks and benefits for themselves.
Thank you, Elizabeth. And yes, Kate, you had a follow-up.
Yeah, I just wanted to emphasize what Aaron said. So often people think of informed consent as a piece of paper that needs to be signed, and it is a process that is ongoing throughout much of a clinical trial and can be quite time-consuming, but the families deserve to have their questions answered and the information that they need to make good decisions. So the process of informed consent.
Right, I agree with you. It's a process. It's not a document, and very, very important distinction to be made here. Thank you, Kate, and thanks, Aaron, and thank you so much, Elizabeth, too. Then, there was a question that was assigned to Dr. Bernard Thibault, and that was, could you please elaborate on the reliability and predictive accuracy of the preclinical models in terms of their translational application? And then follow up on that was that would you elaborate on potential quantitative approaches to optimize dose selection for cell-based therapies?
Yeah, thank you very much. These are very good questions, tough questions. I'm not sure I can answer the quantitative estimate of translational predictivity. I've never thought about calculating this. I don't know if there are algorithms out there for this that could predict the likely success of translational activity. I think that what the systematic reviews show and what people have already realized is that we need more robust preclinical data to inform clinical trial design and whether a new therapy is ready for clinical translation. And so the same rigor that we apply in clinical trials should be applied to preclinical studies. And maybe over time, we will be able to develop such a translational predictivity model. Every animal model has its pros and cons, of course, and it's important to understand what questions we're trying to answer by choosing a given animal model and recognize the limitations. Here I talked only about animal models, but there are some very interesting 3D in vitro models such as lung organoids or brain organoids that are now emerging, and that can also provide some useful proof of concept experiments before moving to in vivo studies, either in small or large animal models.
Thank you so much, Dr. Thibad. Again, I think this question came from Klaus Romero. Klaus, is this answer your question to your satisfaction, or do you have any additional thoughts?
No, yeah, thanks, Bernard. I think that definitely answers the question. I think the notion, as you mentioned, and this connects with what Dr. Siegel presented, the key importance of defining what a given drug development tool is intended to answer and what their applicability is, and then looking at the complementary nature of the different tools that we have at our disposal, and then identifying some of the wherever a tool has gaps. That's not the end of the world. It's an opportunity to identify additional tools that can help address those gaps. So yeah, thank you very much.
Yeah, absolutely. Thank you.
Thanks, Klaus. Thank you so much, Bernard. Then this is a question to basically everybody in the panel, or whoever you think is most appropriate to answer this. For anyone in gene therapy development programs, how do you account for the potential for discordance between the genotypical and phenotypic expression? This is not a question specified to anybody. So anybody who has a yes, please, Deborah.
I think that one aspect of it is dependent on the gene. So, for example, in surfactant B deficiency, some 60% of the mutations are the same. And so when it's presented as a lethal kind of very severe, due to very severe mutation, then it's, the phenotype, there is no discordance between the phenotype and the genotype. But then there are others. So for ABCA3, there's some 200 mutations, and there's going to be some issue with that. And so you're going to have to look for either known pathological variants, or you're going to have to look for null mutations. So I think it's very dependent on your particular gene that you're looking for about penetrance and expression, etc.
Thank you, Deborah. Anybody else? Elizabeth, you had thoughts, right?
So I'll add to that sort of and provide a little bit of the regulatory perspective, and that is the fact that we do recognize that there is a lot of heterogeneity. Some of these diseases, there is very clear genotype-phenotype correlation. Other times there's not, either because it doesn't exist or it's not well researched and is unknown. And so that's one of the reasons that all of these aspects need to be thought about in designing the clinical development program, and it's very much not a one-size-fits-all approach.
Thank you, Elizabeth. Any further thoughts from other panelists? Okay, then going right on to some of the other questions that I've had. There was a question from Stephanie Jones, and the question that what would be the more experienced trialists on the panel expect? That what would be the biggest challenge for engaging families of term neonates with maybe some more unexpected and unplanned participation in research needed compared to preterm infants where some advanced planning or preparation may be possible, at least in some cases. So basically the idea is that do you guys think that is there a difference in terms of engaging families of term neonates versus engaging with the families of preterm neonates? Because in preterm neonates, you already know that you might actually need to get their children to enroll in some of these studies, so you have more time to mentally prepare, but perhaps not quite so much when you're talking about term neonates. Yes, Nicole.
I would just say a little bit, I think it's probably the preparation of the family. So when it's a surprise and maybe it's term, they're not quite as aware, you have to go through the stages of grief rather quickly. So I think that grieving has to occur potentially a little bit faster, getting on board to be aware of a trial. I think speaking of preterm, one of the things to note is that in some of the clinical trials, we excluded very preterm infants with us, the SMA trials in those pre-symptomatic babies. So I think that then once the product was approved, and this is for Zolgensma in particular, there was, data wasn't there for those very preterm infants. So I think that's important to remember as we're going on and maybe moving forward with clinical trials in the future. But just going back to the question of from a preterm infant versus a term infant, I think it's probably that you have time to prepare.
Right. Yes, Kate, you had a point as well, right?
Yeah, I think the other thing that we can do is learn a lot from the newborn screening literature about what it's like to get an unanticipated new potential diagnosis in a family. There's been lots of study done on parental responses and bonding and decision making in that field. So there's lots to learn from there as well.
Thank you, Kate. So, we're rapidly, we do have several questions that are, that have come from the audience that have not yet been answered. We promise that we will get to those questions and one way or another get those questions answered and the answers delivered to you. But, there are 2 minutes and I wanted to give these 2 minutes back to the chairs of the session, that is Mark Turner and Thomas Miller, in case they have any closing thoughts or closing remarks before we go on to this 45-minute break.
Mark, would you like to start?
No, thanks, Tom. Well, I'd like to thank all the speakers for an engaging and concise discussion. I hope this gives us some starting points for some practical discussions about how to distill experience, how to build on the wisdom that people have expressed so that we can make this path easier so that and unleash the potential of cell and gene therapy. So thank you to everybody.
And I could only echo that. We have a number of new participants in an INC conversation that is just beginning. But as folks that have been associated with the INC for some time, I'm sure appreciate our common theme is to make sure that all key voices in the ecosystem are heard and are part of the conversation. So thought leading physicians, nurses, other caretakers, industry individuals, regulators, and of course, parents and families. And that is, I think, one of the points that I'm going to take from this conversation is that's probably of paramount importance for sure on this topic. So thank you very much. Enjoy the break, and we'll look forward to the working groups for those that are participating in that at the top of the hour.
Thank you so much. Thanks, Tom. Thanks, Mark. And thank you to all of our speakers and panelists for their time today. And thank you for the audience for coming in. I think we have like more than 135 attendees in the audience today. And thanks for coming in. We will take a short 45 minutes break and we'll reconvene at 1 p.m. Eastern. Same link, the one that we use for the morning session, where we are going to go over the INC work group updates, because we do have 5 active work groups and they have accomplished a lot in the past 1 year, and we would love to update you all on what they've done today so far in this year. Thank you.
Thank you.